Hi everyone, I'm Ben Tagg, a postdoctoral researcher working for GRIA at UCL. Today, I have the pleasure of showing you around our lab. And here we work on the GRIA family of genes under the GRIA umbrella. And these are important because they provide the instructions to make proteins that operate at the junctions or synapses between neurons, those little cells of our nervous system. Specifically, these proteins allow them to interconnect as complex electrical circuits and give us our behaviour. So today I'll show you how we examine disease-causing genetic variation by recording tiny electrical signals from the proteins of these genes.
Hi there, I'm Ian Coombs. I work at UCL. I am an electrophysiologist, which means I study the electrical properties of cells. Now, when we first find out that a patient has a variant in one of the GRIA genes, the first thing we want to do is to incorporate that variant into our plasmid DNA. And the first step that gets carried out is on this magic box here, which is our PCR machine. And that very easily allows us to incorporate the particular amino acid change that is found in your child into our DNA, so that then we can look at how the electrical properties of those receptors has been changed. Once we've synthesized the DNA, then we can bring it down here into our tissue culture room. We have incubators that grow a cell line called HEK293 cells, and these are cells that just keep on growing and dividing. And we like them because they don't make many of their own ion channels, which means that when we put the glutamate receptors in there, everything that we record electrically is exactly what we put there. So we can very precisely work out the changes produced by the variants. And in terms of what that will actually look like, what we produce from the PCR, well, hopefully I can show you this.
So these are some of our GRIA variant DNAs, just in little tubes inside these boxes. At the moment we're mostly working with GRIA 2 and 4, however we're hoping to incorporate work with GRIA 1 and 3 as the year goes on. So we've arrived here in our sterile tissue culture facility and we're going to be performing a transfection. So this is where we take the DNAs that we need to make an AMPA receptor and mix them with a special reagent shown here on the right in the box that allows them to enter the cells so that the cells can start making AMPA receptors. In this process we also include green fluorescent protein, which allows us to see which cells have taken up the DNA and hopefully made the AMPA receptors. So what we're going to do is mix all of these ingredients together and incubate them. After a period of about 24 to 48 hours of these ingredients being on the HEK cells, we arrive here. So this is our typical electrophysiological setup. And when we look under the microscope at about ×40 magnification, this is what HEK cells usually look like. They have this polygonal shape and they extend lots of processes. However, once we've transfected them, they can look somewhat different. So these are some transfected HEK cells, i.e. they are expressing the receptor DNA that we're interested in, along with a green fluorescent protein. So it's these cells that we'll be aiming for. So now that we have our GRIA variant DNA inside of a HEK cell, how do we assay whether its function's changed? Well, let's see. So on the left here, we see a structure of an AMPA receptor obtained through a special type of electron microscopy that allows us to see atoms within a protein. And what we would usually be able to see is that it is made up of 4 subunits from corresponding GRIA genes, GRIA 1 to 4, as well as some helper proteins like these TARPs. And if you unravelled each subunit of the AMPA receptor, you would see that they can all bind glutamate. And when they do so, as is shown on the right, they open with this rosette-like motion to reveal a tunnel that lets in electricity. And what we do is measure this electrical flow to follow how AMPA receptors open and close, and whether this is different for GRIA variants. When it comes to measuring GRIA-derived electrical currents, we see that they open very quickly, for example on the left. but also on the right. They can actually close in two different ways, and the amount of charge transferred depends on how long the receptors are open, which can often be changed in GRIA genetic variants. So most usually, the synapse has mechanisms to remove glutamate very quickly, and when that happens, the channels unbind glutamate and close in a process called deactivation. So this is shown here on the left. Upon a 1 millisecond pulse of glutamate, the channel opens and closes rapidly. And this electrical wave is what we record. However, when we continue to apply glutamate, as on the right, we have an emergency brake system. You see, we don't want too much electricity to flow into the neuron, or it might fire inappropriately. And this safety mechanism is shown in action on the right. After a few milliseconds, the emergency brakes kick in as the process of desensitization, whereby glutamate has enough time to change the shape of our receptor, forcing it to close. So looking on the right, we can see that if we apply glutamate again and again to these desensitized receptors, it's clear that it takes some time to recover from the use of the emergency brake system. Well, why is this important? In GRIA variants, often there can be a change in the amount of current transferred through a group of open receptors, whereby they either stay open too long and a gain of function, or where they close too quickly and a loss of function. But for now, let's see how we'd assess this in a typical experiment. So Ian places an electrode covered in a polished glass tube under the microscope, and then uses a precise manipulator to control its position and place it in our field of view. So here it comes now. There we go. At this point, he's just moving it down to his hex cell of choice, past another specialist tool. And he turns on the fluorescence to convince us that it has our protein of interest. So we want to extract a small piece of membrane from this cell, hopefully containing AMPA receptors, and for that, visual examination isn't enough. Instead, we turn to a specialist piece of equipment you see it on the left, which looks like something the Ghostbusters would use, called an oscilloscope, that allows us to monitor electrical resistance as we go near the cell. So first, he injects a small current of a fixed size so that he can monitor any subsequent changes of resistance. And you'll see shortly that as he makes contact with the cell, the size of this current decreases as the electrical resistance increases until we have a tight seal. And there we go. So now he's going to apply a small shock to the cell to rupture a portion of the membrane, and we'll see a little bit of leak develop. There we go. So now Ian's about to slowly pull away the electrode to snap a piece of the remaining membrane off, with AMPA receptors on the outside so that they can be activated by glutamate. And this is just happening now. So the membrane has now sealed itself back together and the injected current disappears because the resistance is so high. So here we're bringing the membrane on the electrode back up to the level of another specialist tool that I'll talk about shortly. So this tool is a specially made electrode that consists of two barrels you can see on either side, separated by a septum in the middle. And usually, on one side of this septum, we have a special solution that mimics that found outside all of your cells. And on the other side, we have the same solution but with glutamate. And by precisely applying this for short amounts of time, and at a known time, we can separate out the different phases of how the receptors open and how they close under each program of closure, which is something that's not possible by other electrophysiological techniques. And here you can see the solutions actually flowing, and Ian's about to show us that we can rapidly move the tool backwards and forwards. And this is what we actually see as that tool moves. Glutamate is applied, and a rapid opening occurs. In this instance, glutamate is present for 100 milliseconds, which is long enough to change the shape of the receptor under that desensitization program. So after this rapid downward spike, we see a still fast, but slightly slower closure program in operation. And it's the timing of this closure that we're most interested in, though there are other features to be detected. But for now, let's see a different type of current. So here we're seeing the closure of a GRIA variant, and though it's very fast because we're actually looking at the deactivation here, this variant does in fact close very rapidly. And we can ignore the fact that it's slightly noisier, that's just a property of the cell. But what does that mean? Well, let's find out. So, as you saw, we repeat this recording protocol several times on each patch we pull. And we take the average across time of all of those individual epochs that we saw there to obtain measures of how long a receptor takes to open or close. And doing so, we calculate a few different things, but fundamentally, they just tell us the time it takes for something to happen. So on the top left here, we can see that the particular variant we were recording from just then is open for much less time than the unchanged receptor. And by repeating that experiment across several cells, we can build up a picture of what this looks like. And so, on these top right 3 panels, we can see that it turns out for this receptor, it actually takes longer to open. But it also closes more rapidly by both routes. That is, the receptor closes faster when glutamate is removed, in a short jump as you saw there, but also on the far right top that the emergency brake system kicks in much faster than it should. So the net effect, if we imagine the area contained within these little curves, is that less current is transferred, and so this receptor would be less efficient at making a neuron fire. In our vocabulary, because the amount of current it passes is reduced, we call it loss of function. If a variant transfers more current because it was open longer, we would call it gain of function, and usually perampanel is recommended for those patients. But in part of the work we're doing for GRIA, we recognise that when a variant's loss of function, there's no currently available drug on offer. And this is why it's so critical to be able to target the pharmacology. So, for example, in the work I'm doing for GRIA, I test some compounds called positive allosteric modulators that keep the channel open longer. And we hope that by using them we could transform these loss of function variants into giving the same signal over time as the unchanged receptor. And so on the bottom we can see some example data of using one of these drugs. And this is actually from my positive control, mibampator, which Jeff Swanson has published on previously. So, on the bottom left, we can see that this drug does 2 things for this variant. It completely halts the brakes. Usually the receptor will open and close within about 4 milliseconds. But here it never closes, even if we apply glutamate for 20 seconds. Certainly not desirable. And when we zoom in on the bottom middle panel, we see that the drug also increases the size of the current, changing the nature of its underlying message. So really at this dose, and with this drug, we've pushed the problem the other way. Too much electrical current is now let in, and for too long, even for our GRIA variant. We might predict this drug to be proconvulsant at this dose, but really it at least tells us that the variant responds to this type of compound. So now that we've seen how an experiment works, I thought I might take the opportunity to answer some of your questions. So, what GRIA projects are we engaged with? Well, as well as the exhaustive work that Ian Coombs is doing on profiling changes in function of genetic variants, as I mentioned, I currently work on using positive allosteric modulators to rescue the function of loss of function variants, for which there is currently no approved treatment. And the lab has actually just been funded for another project, looking at alternatives for perampanel in gain of function GRIA disorder. Why is our work important? Well, in terms of its clinical usefulness, most broadly, we aim to understand whether and how a particular variant is disease-causing, and the main classification we generate is gain of function, loss of function, or neutral in that regard. And this information can then be used by our clinical collaborators to understand how varied clinical presentations arise, and whether they're associated with specific signs and symptoms. And this is important because successful treatment strategies and therapies can then be redeployed for patients with variants that similarly alter function. And this does guide clinical decisions. For example, whether a variant is loss or gain of function determines whether GRIA activity should be decreased with a drug like perampanel or increased, for which there is actually no current drug approved. Which brings me to my next point, though. For loss of function variants, though there aren't any approved therapies, the same logic still applies. The outcome of the functional analysis can tell us what therapeutic strategies we should be testing for GRIA to precisely rescue function of that genetic variant's AMPAR receptor. And in terms of loss of function, we've identified 3 types thereof to date, which is slow responding receptors, fast to deactivate receptors, and fast to desensitize receptors. So we hope that by examining the effects of these drugs on different types of loss of function, that's the right way to encourage these compounds towards potential targeted clinical trials. But really all the while we're still grouping patient variants together under different umbrellas of what we might expect them to respond to in terms of future therapies. What obstacles do we face in our work? Well, all our experiments take time because they have a low throughput rate. We suffer from mixed success and high ambitions. So about 20 different independent processes have to go right just to get a single recording. And that, of course, we need many recordings to make up a data set to understand how a change of function occurs. The absolute difficulty of recording is fairly high because of its failure rate. The cells don't want to help us. Sometimes they're unhealthy. Many of them might not even make AMPA receptors, so we can't record from those ones. But often the patch can rupture during recording, and of course with so much equipment, regular maintenance is a necessity. And all of these can combine sometimes to make our technique low throughput. Particularly in Ian Coombs's work, the operation is happening at large scale. There are a lot of variants that enter the lab, with new variants arriving all of the time. We want to be as exhaustive as possible with our work. So, for example, though doing a function analysis experiment is something that can begin as soon as we have a current. If we want to compare a change in function with or without a drug, for example, then the length of time we need in an individual recording, where the cell can't blow up, is at least double. Why is GRIA funding so essential? Well, we don't really have the time or the expertise or the equipment to make our reagents and quality control them. We rely on external companies to give us the tools and equipment we need to succeed. And that's a useful arrangement. GRIA don't want us spending our time fixing electronics. You want us doing science. And in terms of what we routinely use, the equipment cost is quite large in itself. For example, an incubator that the HEC cells need to survive starts at about $8,000, an electrophysiology rig from maybe about $55,000 upwards. But the day-to-day consumables themselves are expensive. To put our DNA into HEC cells costs about $1,300 for 3 months of reagent. And there are many other smaller but more regular expenses like the media the HEC cells need to grow in, glass electrodes, tools. Even the pipettes to fill our electrodes are very expensive. And, we also spend money with external organizations to sequence our DNA plasmids after we've made variants. And, of course, we buy drugs to use on our variants. And all of those costs certainly add up. But really, if GRIA just funded consumables and equipment, our productivity would decrease massively. Contrary to popular belief, pure researchers aren't paid by the university just to be there. We have to apply for funding. And, for example, I'm fortunate to have my salary paid for by GRIA, which means that I can dedicate my entire time to the project and don't have to fulfill obligations to other funding bodies. Which, as well as allowing me to be mission-focused in terms of developing the science, it shortens the project timeframe because these experiments are so time-consuming. And to have a PhD scientist in a major city, I am in the right place to collaborate with other experts with and outside of the field and be where the electrophysiological rigs are. The starting cost is probably about $55,000 in the UK or $60,000 in the US, and that's pre-tax. So I hope that this video has somewhat demystified what a CureGrin lab does and how impactful that work can be for patients. But I also hope that it encourages you to continue to participate in CureGrin. Remember, we're just one of many labs working internationally to cure GRIA disorders. But the work that the Foundation does cannot be understated. By raising awareness, advocating for patients to neurologists and pharma companies, providing essential support to patient families, and by making the science better, CureGrin does everything possible to be the promise laid out in its name. So remember to see the CureGrin website and the GRIA Connect forum for information about getting involved in the latest science support groups, fundraising and events. And we can't wait to see you next time.